Hello, good afternoon. It's week number 9. Gosh, seems like a long time, increasingly. Okay, so where are we at? We have spent the last week, I think it was last week, working through the analysis of our RNA-Seq data. Particularly, particular interest to me today, anyway, is the fact that we split our dataset in two because we did a PCA. I can't even remember how long ago now. I would have to look it up on the channel myself to figure it out. But we did a PCA. I'm guessing a couple of weeks ago now. Let's see if we can find out actually, because I think it would be useful to know exactly how long all of this took us. Here we go. Playlists. That one, I don't want to play it, I just want to look at it. There we go. Right, PCA was number 31. So, what are we at now? Number 38. So that was not the beginning of last week, but the beginning of the week after. No, the week before, sorry. So yes, so about 2 weeks ago, we did PCA, and as a result of that initial PCA that we did, we decided that we would split our dataset in two, or rather, I decided that we would split our dataset in two, because it really did look like two separate independent datasets from two different cell lines. So we split the dataset in two, and then when it came to working our way through differential expression analysis, we did that exclusively on one of those two datasets, that dataset from the NHBE cells. And we worked through that to get results, and that's where we got to. By the end of last week, we had differentially expressed genes in that cell line, and then we looked at some plotting of those genes. So we made volcano plots and we made MA plots. That was in number 34, and then so number 34 was the beginning of last week, I think, or maybe Tuesday last week. So we got differentially expressed genes on Monday last week. We looked at plotting on Tuesday. Then on Wednesday we looked at annotation last week. And Thursday we drew heat maps, and then on Friday we looked at pathway, or a little bit at pathway analysis, or particularly gene ontology enrichment, really. But we did look at enrichment analysis without going into too much depth about sort of more general pathway analysis. And what I thought that we could do this week to mainly to kind of reinforce a lot of those messages and a lot of the lessons that we drew from those. What's that? Since PCA, we've done 1, 2, 3, 4, 5, 6. So this is the seventh session since then. So there was a lot of material introduced across those sessions, and I thought to reinforce all of that material, and to make sure that we've not just done it once and then forgotten about it, I thought we would effectively redo all of that work, but redo it in the second cell line. So this serves two purposes. One is just that pure reinforcement of the lessons that we've learned over the last week and a half or so, and making sure that we can apply those same lessons in another data set, even though it's very closely related. But the second is also to give us a point of comparison, to be able then to go on later this week and ask the question, are these two cell lines behaving in the same way when they are challenged with this virus? Is the response of the NHBE cells and the other cell line, who I'm sure I'll burn into my memory over the next few days, the 40 something, I think, cell line, are they essentially responding in the same way to this same challenge? Do we see similar or the same differentially expressed genes? Is the general change in gene expression, is there concordance in that change in expression? So that's what we're going to do. That's my goal for the next couple of days at least. I think a couple of days to repeat the analysis, and then hopefully by Wednesday we'll be in a position to think about comparing those analyses. So comparing the analysis of the NHBE cells with the analysis of the other cell line, A549, that's it, the A549 cell line. And using that comparison as another as the next step along our road in learning facets about R and about bioinformatics more generally. Okay, so that's my plan for the beginning of this week. I think that should at least take us through until Wednesday, and then we'll see where to go from there. So this is the script as we've written it so far. So we're up to a couple hundred lines, nearly 300 lines of code. What I'm going to do for the second data set is I'm going to start a new script, and then we'll have a third script for the comparison once we get there. The one thing that I want to do before we leave this altogether is I just want to show you how to write your results out to a table, because I think that's probably quite useful. So let's just do our actual differentially expressed genes. So filter DF3 is our 141 differentially expressed genes. Let's write that file out, that object out to a file. And so if you remember, when we read datasets in, we used read CSV or maybe read TSV to read datasets into R. We're going to use write TSV to write files back out again. So write _TSV. This also comes from the reader, despite the fact it's a method for writing data out. It comes from the reader package. It comes with pretty sensible defaults. So normally you can just get away with saying, well, the thing that I want to write out is filter df3. So that's your first argument, and then the second argument is the place you want to write that to. So I want to write this out to. Because I'm in my project. I'm in this COVID RNA-Seq directory, and you can check that by going into your files. This usually opens in your current working directory. And we can just check in R that what our current working directory is with get wd, and that will print your present working directory. So wd is working directory, the same as it was at the command line, and that tells me that any files that I'm going to write, sort of by default, will end up in this directory because this is my working directory, and that's a perfectly fine place to put them. So let's make a file called filtered NHBE results.txt. So this is a text file that is the results of our first differential expression analysis. This is quite a useful thing to be able to do, because obviously we can do lots of things with these results in R, but it can be useful to take those results to other software elsewhere. Maybe you want to upload your results to Ingenuity Pathway Analysis or some other piece of software for doing that kind of follow-on work. So once we've written the file, you can see that it appears in our file system down here. RStudio shoves it at the bottom to begin with, just when it's new. If we refresh our listings, you'll see it pops into the right place in alphabetical order. So that's our our txt file that we've just downloaded. So the one other thing that I wanted to show you with this, because I think this is something which is really important, is the quote-unquote correct way to load this file into Excel, because this is a table of data, and I know that when it comes to working with tables of data, lots of people like to work with them in Excel, and I completely get that. I understand that Excel is a useful platform for viewing and working with these kind of tables. So I just wanted to not completely ignore that fact, but to show you the way that I would try to work with this kind of table of data in in Excel. Sorry. So I've opened Excel and I've gone to open a file. So Control O or then go to File and go to Open. And I'm going to navigate to my text file that I just wrote here. So this is the text file that I just produced from my R session. And actually looking at this, it looks like my file that I've produced doesn't have the annotation in. So that's not particularly useful to show you, so let's cancel that. Let's go back and let's add the annotation into this file. I remember I did that last week. I filtered it, didn't I, with the annotation. It was called here, anno df3. That's the data frame I want to write out, not filter df3. Apologies. So write that back out. That will overwrite that file. You'll have no qualms about overwriting that file. The size of the file has gone up. It was 16 kilobytes before. Now it's 34 kilobytes. And that's because I've written the annotation into it. So let's try again to open this file in Excel. So here's my Excel file dialog. So I've just clicked open and I've navigated to the file on my Mac, and I'm going to point it to this file, and this time you can see that it's got the annotation in it. So in this little preview window that my Mac gives me, you've got this little preview of all of the data, so we can see that the data is in it. Okay, so we're going to open this file. Now, to lots of people this would seem like a fairly trivial operation, but actually with the way that Excel operates, you have to be slightly careful about importing data, especially data about genes, into Excel. So I just thought it's worth just showing you taking that care with this data. So here's the data in this sort of preview form. We know that it's delimited, so we asked for a tab delimited file because we wrote a TSV. So we asked for a tab delimited file, so it's delimited data. So we tick that option on this first page. Then we step through it. It shows us now the table separated by those tab characters. So we can just check that it's got the table correct, and we can see that that's previewing quite nicely. So then the next screen is the really important one, and this is the types of columns that you want Excel to import. Now Excel will by default set every single column to be general. And for most of the data that you import into Excel, the general data format is absolutely fine. The one thing that really breaks with the general data format, actually there's two things that spring to mind. One thing is the way that the barcodes are formatted for methylation arrays, and if we ever talk about methylation arrays in more detail, maybe we'll come back to that. And because they're long numbers, they get turned into scientific format, and that causes rounding errors. So they stop being precise numbers and they become just numbers that are in the tens of millions, and they get rounded. That's a disaster, which I've had bite me on occasions. But the other thing that really suffers from being formatted in this general data format is gene names. And gene names and I'll show you an example of this in a minute, but gene names can be changed by being formatted in the general format. So when you import gene lists, especially into Excel, you need to make sure that you tell Excel that your gene name column is a column of text, not a general column. And I'll show you why that's important in a minute. Okay, so you need to make sure that you tell Excel that your gene names are text. Okay, so then we're going to finish, and that will import that data into Excel. So there we are. That's our data imported in, and our gene names importantly have been preserved by that process. And you may not necessarily notice superficially, if I imported these as general, you may not have superficially noticed any difference. And with an awful lot of gene names, there isn't any difference with between general and text. But I'll show you an example. So, okay, the decapentaplegic genes. So they're called, their abbreviation, their gene names are all DECs. So DEC1 is a gene, and if you format DEC1 as text, that didn't format as text. That was interesting. Then it would be DEC1. But if you allow Excel to format it in its general way, then it gets changed into, you'll see DEC1 gets changed into the 1st of December 2001. So Excel has guessed what type of data it was that you entered, and it's guessed that it's a date. And Excel is extremely keen to change things into dates. Really keen to do it. And so there's the DEC genes, there's the sept genes, so the septins, they get changed. There's the March genes, they get changed into dates. There's also a family of oct genes. I can't remember what their gene names are, but the oct genes get changed into dates as well. There might be some others. But lots and lots of gene names. There are plenty of gene names that get changed into dates. And you might think, okay, well, this is fine. I inputted this as text, so I will just change it back to text, and that will all be fine. It will preserve what I wanted it to be. So then you go into your format for your cell, and you say, well, I want this cell to be text. Okay. And magically, when you convert a date, something that Excel has converted into a date, back into text, Excel gives you the underlying data representation of that date, which is a floating point number. So this number is the number that Excel uses to represent the date. So the date in Excel's data model, dates are saved as numbers and then converted back into dates in the display layer. So again, you may not think, okay, well, fine, this has changed this into a number, but that number represents the date that Excel thinks was entered at the beginning. So I just have to find out what number that date is represented by, what date is represented by that number. And then I can change it back into the date into the gene that it was before, and this time I'll enter it as text, and it'll all be fine. Obviously this is a very deep rabbit hole, and I could keep going. The one thing, my last kind of point about this is that number that is used to represent the date by Excel under the hood is different in different versions of Excel and on different platforms. So if I've created this Excel file in whatever the latest version of Excel is, the Office 365 version of Excel on a Mac, the number that date is represented by that the date that is represented by that number will be different to the same number as if you'd have used Excel 2011 on a Windows machine to generate this data, to read in this data. The underlying numbers on those platforms are different. And so actually the process of converting from the number back to the original date is much more complicated than you might initially think. And so if you import data into Excel, and I completely understand why you would do that, and what I'm not here to do is to tell you not to do that. I'm not interested in stopping people or asking or sort of trying to get people to stop using the tools with which they're most comfortable. And I get that Excel has a lot of uses for lots of people who are very used to it as a platform. But just be aware of the way that it treats your data, of the things that it does to your data, and allow for that. So open the file in a particular way, which prevents these things from happening. So actually with this data set, scanning through this data set, I'm just going to delete those things I added. Scanning through this data set, there doesn't appear to be any problematic gene names in this particular list. Okay, it doesn't look like it's too problematic. But the way that I opened it, by opening it with that column as text, I will have prevented any gene names from being changed anyway. Just quickly, the other thing that I like to do with data once it's in Excel is format it as a table, because that's quite a nice way of getting the data formatted so that you can just make the columns the right width, so that you can then filter your columns on various things if you want to. So if you had your whole gene list in here, you could filter out on the adjusted p-value. If you for some reason you're only interested in genes on chromosome 3, right? I don't know why that would be, but maybe that might be the case, for instance. Then you can use the tools, the table tools in Excel for filtering nice and quickly. It's quite a useful tool for that. It's quite a nice little table viewer in lots of respects. It's a slightly heavyweight for it, and it's not what it's intended for, but it does work quite nicely for that. So I know that's a bit of an aside, but it kind of occurred to me as we started today that we never actually got round to saving our data to a file, and that might be something that we want to do. Okay, so I'm going to save this script, and I will upload this as the script for the NHBE cells to YouTube, to GitHub. This is YouTube. GitHub is another service. So that's yes, that's kind of the finishing point, as far as I'm concerned at the moment anyway, for NHBE. And what we want to do now is repeat this analysis for the A549 cells. So in order to do that, I'm going to start a new script, but I am going to copy and paste some of this stuff across, because things like reading in the metadata, we're going to do in exactly the same way, but I want the script to be self-contained, so I want to make sure that we're doing the same things in the same scripts, but my script is self-contained. Okay, so let's open a new R script, and then let's copy some stuff across. So let's copy this across, because this is useful. And then, okay, that gets us to a sample table with 12 rows. So let's copy that lot across. Copy and paste, first 22 lines. And then this is our sample file. So let's do that as well. And our gene map. Paste those across. But then we don't need to pull in all of that. We need to divide the data set up before we read it in, which we did here. This was it. So I'm just going to get to that point. I'm going to copy all of that, and then we're going to change this as we go. Okay, so rather than rewriting all of that code, I'm just copying and pasting it across, and then we're going to edit this to be appropriate for our new set of data. So we're going to do the same package imports because we'll end up using the same packages eventually. Then we're going to read in the same sample table. We're going to make the same sample files vector, the same gene map, but then we want to, instead of doing 1 to 6, we want to do 7 to 12. So that's the second set of 6 samples. And it, instead of being count data NHBE, it's A549, which is the second cell line. So we need to change our names as well. So wherever we see an NHBE, we need to replace it with A549, which we can do with search and replace. So I hit Command F there, but you could go to edit and find, and that brings up this little search and replace dialog. So we can put in NHBE in here, and then we can replace it with A549, and we can do that everywhere. So we can just click all, and that will replace all of those occurrences. So that does that for me. And then, so that's sample files 7 to 12. We also want the 7th row to the 12th row of the sample table. And we want to, okay, we'll come to this in a second. Okay, so that's everything that we're going to do in the immediate, at the beginning of this anyway. So, okay, what I'm going to do is I'm going to save this script in progress as COVID A549 analysis, and I'm going to quit my R session, quit RStudio and restart it so that we can walk through that process a little bit, because I know some people have had some issues in sort of recovering their R session after they've restarted their computer and things like that. So I just want to show you the behavior of RStudio when you quit it and restart it, basically. So if you remember, I am in my project here called COVID RNA-Seq, which means that my working directory is not my home directory, but is my project directory. So this is the directory that my project lives in. When I quit RStudio, so when I press Command Q, or when I go to File, File and quit RStudio, sorry, my RStudio menu, and quit RStudio. So when I quit the application, RStudio will prompt me as to whether I want to save my current environment. And the place where it will save my current environment is a file inside my my project directory, inside this COVID RNA-Seq directory. So I'm going to quit RStudio, and it's going to prompt me, do I want to save my workspace image to this file? Okay, so home/lockdown-learning/COVID RNA-Seq and the file is called .rdata. Okay, you can save it or not. It's up to you. What will be saved to that file is everything that's in your current environment. So all of these things that we've created, and you can see the environment's got quite big and quite complicated now. All of these things in your current environment will be saved into that .rdata file. You can achieve this inside your current R session by clicking Save in the environment. You can save this environment to a file by clicking that and saying, okay, well, I want to save, this was my NHBE analysis, and it will save it by default as an RData file. Or we can be explicit about it and say .rdata. Okay, so that will save that file. That's gonna, so this, if you can see what it did in R is it ran this function called save.image. And so this is a function that you can use within your R as well to save a snapshot of your current environment into a file. If you want to save a single object, a single variable into a file, there's a function called save, which will do it as well. So save with a single variable or with a list of variables will save just that variable or variables to a file, whereas save.image will save your entire environment. So what you'll see is that file is quite big. Okay, so this NHBE analysis RData file is about 100 meg. So it's a reasonably large file, but that's my current environment. And that's what will be saved to your .rdata file if you say to save it there when you quit RStudio. Okay, so if you answer save to this, then it will save your environment into that file, and then when you restart this project, which should happen by default when you restart RStudio, it should reload the last project you were in. But when I restart this project, that .rdata file which is being saved will be reloaded. So my environment should be restored. Okay, so I'm going to save it just to show you that happening. And so RStudio is going to quit, saving that file, which you can see takes a little while, and then RStudio is going to close down, and then I'll restart it to show you the fact that so because I saved that environment, that will be restored once my RStudio restarts, and I will have all of those things back again. So it's just taking a few seconds to save that .rdata file. I'm just going to show you it in terminal actually. So pop my terminal up and cd into my project directory. So this is my project directory, which is in the shared directory with my virtual machine, if you remember setting all of that up 9 weeks ago. So in here we have a number of files. What you don't see in here, incidentally, is that .rdata file, and that's because it's a so-called hidden file. And you can see all of your hidden files in a directory by adding the -a option to ls, and that lists all of your hidden files in the current directory, which includes my .rdata file. So this is that file that's just been saved when I quit RStudio. There's also some other R files which are specific and hidden as well. So R history, as the name suggests, is the history of your R commands that you've run in this RStudio project. And then this directory, which is also hidden here, rproj.user, that's a hidden directory, and that's to do with the project setup for this particular RStudio project. So I'm going to reopen RStudio. So here you go, RStudio is opening back up, and what should happen is that it should open with the project that was running when you closed it down last time, which it has indeed done. So it's opened up with this COVID RNA-Seq project running, and it should then resume your state. So it should load back in the things that were loaded when you closed it down. So it should load in the scripts that you had open. It should load up your environment as it was, and which it has done. So you can see it says workspace loaded from home/lockdown learning, blah blah blah. What it hasn't done, so we've got a whole new R. So we've got the R, all of the R prompt up here, which is printed when R first starts. What it generally hasn't done, with a few exceptions, is reloaded the packages that we had loaded in. So your packages will all have been detached, and we'll have to reattach our packages. But it has reloaded, so it's all of the data sets that I had open have all been reopened in View, and my scripts are still here. Although, weirdly, not the new one. Oh yeah, the new one is also there at the end. And my environment has been repopulated with all of my old environment. Okay, so if when you'd have quit, and you'd have said, don't reload my environment, don't load all that stuff back in. If you'd have taken a snapshot like I did before, you could load that back in with load. So load and then an R data file. So we called it, I can't remember what we called it. That's what tab complete is for. There you go, nhbe analysis.rdata. That command in R would load our saved environment back as well. So that would be the other thing that you could do to reload your environment if you hadn't saved it in your .r data file. But as it goes, I'm going to clean my environment up. So I'm going to sweep it clean. Yes, I do want to remove everything. I've got clean slate of packages because I've started a new R session. So all of my packages will need to be reloaded. All of my plots should have disappeared and various other things. So we're going to start again at the top of this script. So this is our, let me close down all of these data sets as well. We don't need those hanging around. Okay, so here we go. Let's load in our packages first of all. So just going to highlight all of the lines that I want to run, and then press Command and Enter or Control and Enter, and that will reload. That will load in all of my packages. So that's reattached all my packages, and now I want to read in my sample table. And actually, this is just another way of doing the same thing. And then unique there reduces you down to 12 rows. Okay, so our sample table now is 12 rows long, and it has all of the NHBE samples and all of the A549 samples in it. So we want to select out just the 549s from that. We're going to make our sample files. Actually, let's do this a different way. Let's filter here. So sample table. So we've been filtering in square brackets as we've gone along. So the way that you filter using square brackets, I just want to show you that these two things here are equivalent. So if we look at a sample table, we can say cell underscore line with a capital C and a capital L. So sample table dollars cell underscore line equals A549. Okay, and we're filtering the rows. So our statement for our filtering. So this is a relational expression that will evaluate for true for the samples that we want to keep, and false for the samples that we don't want to keep. And we want to filter the rows, so we put that relational statement before the comma, and we want to keep all the columns, so we put nothing after the comma. So this will filter that sample table down to just those 6 rows that we want to keep. Okay, so that's what we've got here, the 6. You can see it's a tibble which is 6 by 6. So it's got 6 rows, 6 columns, And importantly, in the cell line column, they're all A549. Okay, so those are the 6 that we want to keep. The other thing that I wanted to show you was the dplyr way of doing this. So dplyr, which is from the tidyverse, also gives us ways of filtering data frames, which can, to especially to new users, look much easier. So the arguments for this are the tibble that you want to filter as the first argument. So we want to filter this thing called sample table. And then the second argument is the relational expression by which you want to filter it. And in this instance, we don't need to use this arcane syntax for pulling out a particular column, because dplyr knows about the columns. So it knows that cell underscore line, it knows what we're talking about here is a column of the tibble in the first argument. So, and now again we want cell line to equal, so equals A549. And so this line of code here achieves exactly the same as this line of code. The results are the same. So there you go, a tibble which is 6 by 6, which has all of our A549 samples in. But I think, I know I've been doing this all the way along, and you kind of come to these realizations that maybe you shouldn't have been. But I do think that for beginners especially, this is easier to understand. So we've got a thing called sample table. It has a column in it called cell line, and we want any rows from this table where the entry in cell line equals A549. So that's what we're going to do to do our filtering. So this is going to be our sample table A549 equals that. And then our sample files A549 equals the same as we've done here, but sample table A549. So we're going to pull that column, pull the sample name column out of this table that we just created here. And we're going to add to that this text /quant.sf. Okay, so this gives us our vector. Oh, I need to actually run the line above. This gives us our vector of paths to our quantification files, one for each of our 6 samples, so a vector of length 6. So hopefully people remember doing this the first time around. I'm trying to make this slightly simpler this time around. So again, we can pull those sample names back out as well to be the names of that vector. The reason that we do that is because the names of that vector via tx import follow through to be the names of our columns in our count data. So we need to name the vector that we're using here. The gene map is the same. Okay, so the gene map is exactly the same as it was before. Nothing's changed about that. We've already done this bit. And now we just do this bit. Okay, so done this bit as well. So our count data is read in by tx import, which we've attached in our package, in our library statements at the top. So tx import is already available to us. We've got this vector of sample files, which we looked at here. They're of type salmon, so they come from, that's the program we use to generate them. We've produced our gene map, which, if you remember, is this 2-column data frame, or tibble in this case, with the first column being the ensemble transcript IDs and the second column being the corresponding ensemble gene IDs. And we want to ignore tx version because we've got the dot and the version on the transcript IDs in our salmon results files. So we will run this to read in our count data for our 6 samples. Okay, and now we want to set up our conditions. So if we look at our sample table, let's do this with view. If you remember, okay, let's make this wider. Here we are, that's our 6 columns. So in this sample table, the description of what has occurred with these samples is described here in this data. This treatment column describes the conditions of our experiment. So 3 that have been mock treated, 3 that have been SARS-CoV-2 infected. But we want that description to be slightly simpler. It's better to keep it, to have no spaces in it, for instance, just because in the long run, in our having no spaces in as many things as possible really makes a lot of sense. We don't want spaces in column names if we can avoid it. We don't want spaces you can't have spaces in variable names, and that's kind of part of the root of all of this, is not being able to have spaces in variable names. So we want to make a simpler description of this treatment where we just have mock or infected. And the way that we set this up was to add a factor, because we're describing a variable which can only take particular levels. It's describing our experiment, so it can only be mock or infected. It can't take an arbitrary third value or fourth value. So we want to describe that setup as a factor. And importantly, for what we do next, for DESeq, we want that factor to be in a particular order. So for DESeq, it's important to have, in our factor, to have the base level of expression, what you would normally think of as the control, as the first level in the factor. So let me walk you through this a little bit. If we just let's break this out into separate bits. Okay, so here we go. So why did it do that? We know that we want a vector of length 6, which describes our 2 conditions in our experiment. So the first 3 are mock, and the next 3 are infected. Okay, so this call to rep will do this. It will give us a vector which has 3 mocks and 3 infecteds in a row like this. So mock, 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 infected, infected, infected. Okay, if we just turn this into a factor as it is, so this is our so let's call this let's just turn it into a factor by, okay, so if we just turn this into a factor as is, factor this, then the levels of the factor will be set in alphabetical order by default, because the vector that the factor is being created from is a character vector. Your levels of your factor end up in alphabetical order. So we've got mock, 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 infected, 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 and the levels are in alphabetical order. So infected is the first level and mock is the second level. Now when you load this into DESeq, which is what we're doing next, DESeq will treat the first level of the factor as the base level of expression, the comparator level of expression. So it will subtract the infected expression from the mock expression when you do your differential expression calculations. So what that means is it will look like when you have positively regulated genes, so positive fold changes, what that means is that the infected is lower than the mock because you're subtracting a smaller number from a larger number, so the mock expression is higher, and that intuitively is the wrong way around. What you would expect to say is if a gene is upregulated, so if you've got positive fold change, you would expect that to be higher in your experimental condition rather than your control condition. So you'd expect that the level of the expression of that gene to be higher in the infected samples, not in the mock. And so what you want to do is you want these levels to be the other way around, so that DESeq can deal with this properly and in the way that you expect it to deal with it. And that's what this levels argument to factor does. The levels argument here is saying, okay, I know that the levels are infected and mock, but this is the way round that I want them. I don't want them to be in alphabetical order. I want them to be in the order that I tell you I want them to be in. So I want them to be mock first and infected second. And so if we run this command, then you'll see that the factor is exactly the same. So mock, 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 infected, infected, infected. So the factor is exactly the same, but the levels of the factor are the other way around. So rather than being infected mock in alphabetical order that they were before, instead in this case they're mock infected. So they are the correct way around for DESeq to then analyse them. So, okay, there we go. That creates our conditions column in our sample table. So if we go back to our sample table and have a look, then you'll see that that column has been added. And what we're going to do, I think, just to finish off, because I see some people saying in the comments saying they can't catch up and can't keep up, and also I see my viewer numbers going down precipitously, so I'm guessing I've lost some of you today. So I'm going to wrap up. But so now that we've got that data, now that we've got our sample table subsetted in the right way, we've set up our factor in the right way, we've imported used TX import to import our data, then we can use DESeq to read that data back in. Okay, so this is going to read in the count data here that we imported via TX import. We're going to set the col data to be our sample table, so this is the data that describes the columns, and then we're going to use that factor that we just created to describe the experiment in the design. So that will set up our data. And what I'm then going to do, I'm going to delete all of that. So this is my final step for today, is I'm going to use this method DESeq over this data set 549. There we go. So this DESeq function here does everything that the three functions we used last time do. So DESeq here does it does estimate size factors, then it does estimate dispersions, then it does n binom wild. Is it wild test? Remember now. Yeah, and binom wild test. So it does all those three things in one go. So it's a slightly more convenient way of doing this, of doing these three things. Just saves you a bit of typing, really. It's not doing anything differently. So there you go. It tells you what it's doing. So estimating size factors, estimating dispersions, which is then the next few things, and then fitting model and testing. That's the end binom wild test. So it's done those three things in a single, in one single function, and got us to a point where we've effectively analysed our data, and now we just need to extract the results from that, which is what I'm going to do tomorrow. Okay, so at that point I'll call it a day today, and then tomorrow we will repeat pulling the results out from this and reproducing some of the plots that we made last time, so that we can have a look at those results and see if they differ significantly from the NHBE results. So that will be tomorrow. So, okay, so I'm aware that today I've probably gone quite quickly. And I kind of did it intentionally because I'm slightly retreading old ground, because I'm aware that we've done this before, and I don't want to spend days and days and days redoing things that we've done before. I also want to demonstrate that once you've done it once, it is possible to ramp up it gets more fluent. You've done it before, you've got the code. Copying and pasting now becomes your friend, right? So you've written this code before. There's nothing substantially different about this code this time. But as long as you take it a line at a time and make the changes that you need to make as you go along, then it should be relatively straightforward to write the same code the second time for the second example. That's kind of my reasoning for going a bit quicker this time around. And I'm sorry if anybody got lost. But again, I will upload the code. It will be available on the GitHub. And obviously you can go back over these videos if you want to, if you can bear listening to my voice for an extra hour. Although you are still here 36 and some hours later, so maybe I shouldn't judge. Okay, right. So with that, I'll call it a day for today. So come back tomorrow for the second retread of retrieving results from our DESeq dataset and drawing our plots as well. And also we've got to retrieve the annotation as well. Don't forget that. So we've got to look to Biomart to retrieve the annotation for this data, and we'll make our volcano plots and our MA plots as well. So that will be tomorrow. And then on Wednesday, we can really compare the results for the NHBE cells with the results of the A549. Okay, right. See you later. I'll stop talking and sign off, and I'll see you later. Thanks for tuning in. Stay safe, keep well. See you again next time. Bye.